I'm Isacco Beritognolo from CNR, Italy. I'm here to present the results of a genomic study on the natural resistance of Castanea sativa to the Asian gall wasp, Dryocosmus kuriphilus. It represents, sorry, for slide. It represents the most invasive insect pest for chestnut. In the last decades, it has become a major threat in Europe after its first introduction in Italy in 2002. And now the only available control strategy is the massive release of its natural parasitoid Turimus sinensis. This control strategy has proved to be effective over long term. And most of the research has been focused on the control strategy, and the species Castanea sativa is generally considered highly susceptible to this parasite. Previous study observed a wide range of variations susceptibility to the gall wasp in the cultivated germplasm, but the natural resources, genetic resources of Castanea sativa are mostly unexplored as source of resistance. Therefore, the objectives of our study were first to evaluate the variation in resistance to gall wasp in European provenances of natural Castanea sativa. And second objective was to identify genetic variants and candidate genes associated with resistance. The study has been carried out in common gardening trial located in central Italy with six provenances from Spain, Italy, and Greece. And this plantation was established in 2002. So it contains adult trees, and about 1,000 plants are present now. And preliminary survey of gall wasp infestation was carried out in the years 2014, 2015, and we observed that the Greek provenances Pico and Orsiatis had the lower infestation, mean infestation rate, and interestingly, a higher number of plants with a low infestation rate, an infestation rate lower than 10%, in particular the provenance Orsiatis that you can see on the right. Subsequently, two other independent studies conducted by entomologists confirmed our observation, and all these results show that the provenance Orsiatis expressed the lowest susceptibility to gall wasp and a high proportion of plants with very low infestation rate or immune plants. So this plant material represents a good model to investigate the genetics of plant resistance. From the highly skewed distribution that we observed in the provenance Orsiatis, we speculated that a major genetic factor is present in the resistant plants of this provenance. And to test this hypothesis, we set up a genomic experiment based on the principle of the bulk segregant analysis. So we designed a genome-wide association study with two pools, a DNA pool from 25 plants with extreme phenotype of resistant plants, and the DNA pool of 25 plants with extreme phenotype of sensitive plants. And we expect that the differences in allele frequency between these pools will identify the genetic variants associated with resistance. Then the two DNA pools were sequenced on an Illumina platform with a depth of coverage 50-fold. And after processing of trimming and filtering, the sequence reads were mapped to the last assembly of the reference genome of Castanea mollissima. Then the software package population 2 was used in that analysis to compute FST analysis between the pool on sequence windows of 1 kilobase, then to estimate the differences in allele frequency between the pools, and then to determine the significance on these differences by a Fisher test. So in the first step of that analysis, we consider sequence windows of 1 kilobase to perform FST analysis between the two pools. In order to identify the genomic regions of maximum divergence between the resistant and susceptible plants, we analyzed the distribution of the FST value on the whole genome, and we fixed a threshold, you can see here, to determine the outlier FST values. In this way, we identify 28 sequence windows with a local increment of FST, which corresponded to the divergent genomic regions between resistant sensitive plants. And these regions covered 13 contigs and included about 400 single nucleotide polymorphism. In mapping the reads, we have to consider that the present assembly of the reference genome is not structured in chromosomes, but the contigs are anchored to linkage groups. So we carried out the analysis of FST distribution for each linkage group, for all the linkage groups. And this step highlighted the linkage group K with the FST distribution shifted towards higher values than the other linkage groups. And looking at the plot of FST mean along this linkage group K, we found 7 contigs with a higher FST, with local and sharp increment of FST value. All of them concentrated in a small region of about 11 centimorgan. And the next step of that analysis was focused on this target region. So after FST analysis on the sequence windows, we identified the significant differences in allele frequency on individual SNPs in the target region of the linkage group K. For this, we fixed a stringent threshold represented here by the red line to identify the single nucleotide polymorphism strongly associated with Gall-Wasp resistance. In this way, we found 42 significant SNPs in 3 contigs. Here we show the magnification of each contig with the plot of the significant SNPs over the red line. The gene model annotated, and gene models annotated in the reference genome. These 42 SNPs were both intergenic and intergenic. Here in the first contig, one SNP is located within a gene encoding a protein of the cytochrome P450 family. And 2 other SNPs are very close to other copies of the same gene. In the second contig, 28 SNPs are within or nearby 2 copies of a gene encoding another member of the same gene family of cytochrome P proteins. Here in the third contig, the significant SNPs marked a gene encoding another protein of the cytochrome P family, and a gene encoding a GTP binding protein called RAC or ROP. And in total, the intragenic and the intergenic SNP identified 4 different genes in this target region that were present in single or multiple copies, and there was a prevalence of copies of genes encoding family of the cytochrome P450 family. So the preliminary, the conclusion that we can draw from our very preliminary results are that first, the linkage group K is likely involved in the resistance to the gall wasp. Actually, our results are consistent with the previous study, a mapping study that identified a major QTL, explain a large part of phenotypic variance on the same linkage group of the hybrid cultivar Boucher de Bédizac. This is an hybrid. Second, the significant SNP divergent between the two pools help us to identify the candidate genes and the flanking regions that may be associated to regulatory sequences. In our first annotation, we identified some candidate genes for resistance that belong to two gene families. The first group includes some members of the family encoding cytochrome P450. These proteins are known to be involved in the metabolism of various compounds, such as hormones, defensive compounds, fatty acids, and many others. The target region of the linkage group K is very rich in several copies of this gene family, but at the moment their role in plant-insect interaction is unclear, is not well defined. But the other candidate gene belong to the ROP gene family, also called RAC in animals. This gene seems more directly related to plant-insect interaction, as it is known to be involved in the defense of cotton plants against insects. So it is a good candidate. The short-term perspectives to complete the study are first to improve the annotation of the target genomic regions identified by the characterization of the synonymous and synonymous polymorphism in the intragenic SNPs, and the analysis of the intergenic SNPs can help in searching the cis regulatory elements and the promoters of the candidate genes. But in order to confirm that these genomic regions control gall wasp resistance in the species Castanea sativa, the results require a validation, a biological validation by further genetic experiments on different and related plant material, such as other provenances, natural populations, variety, all plant material that should express a variable degree of resistance. The identification of the genetic basis of resistance will improve the overall comprehension of the interaction between gall wasp and chestnut, and may assist the genetic improvement and also the development of effective methods to control this parasite. I would like to thank all the members of the genetic laboratory team of my institute. I would like also to thank Dr. Angelo Massacci, as former director of the institute, for funding the initiative to support internal projects proposed by non-permanent staff. And Dr. Romina Caccia was one of the entomologists who carried out the field observation for her relevant help in selecting the plants and making the experimental design. Thanks for your attention.